and welcome to another episode of 3-Minute Histology with Jamie Chapman. I'm Jamie Chapman, and we'll be looking at the small intestine here. Now, this is used as a special stain called Alcium blue. We'll talk about what that means once the 3 minutes starts. Okay, so Alcium blue is a stain specific for acidic mucopolysaccharides. Basically what it does is it highlights acidic mucus within this section. So these little dots, these little blue dots or green dots represent goblet cells. So this material here is actually mucus covered, looks like plant material here. So let's have a look at this in a little bit more detail. So as we zoom in, we can see the distribution of the goblet cells amongst the epithelium of these villi. So these are villi. You can see a villi here with its loose connective tissue in between. And as we zoom in, we can see these blue or green Alcium blue stained portions of the goblet cells. So what the Alcium blue is staining is the mucus granules at the apical portion of the cell there. And you can see them being extruded on the surface. So goblet cells are unique within exocrine glands because they secrete via a process called compound exocytosis. So rather than secreting each individual secretory granule, they actually secrete them en masse, all together as one big loop. So they go through about 2 secretory cycles during their lifespan of about 5 days, and so they just release all of their mucus onto the surface in one big loop onto the surface there. So these are our enterocytes. So these are the nuclei of our enterocytes here, and so the cytoplasm's here. Here we've got the lamina propria. If we scroll down, we can actually see the base of the intestinal glands. So I might just zoom out a little bit just to give you a bit of a better idea. So you can see these intestinal glands extending down into the base of the intestinal crypts or the crypts of Lieberkühn, and you can see that the goblet cells originate from the base of these glands, and then eventually they migrate their way back onto the surface of the villus, the small intestine epithelium there, and then eventually they're lost from the surface there, and they get replaced continually through mitosis from those intestinal glands. So you can see them scattered throughout there. These little dark bodies represent mitosis. So we've got lots of mitosis happening at the base of these crypts of Lieberkühn. And then down here, we've got a little bit of evidence of some connective tissue. And then we've got our muscularis on the outside. Anyway, I thought that was quite an interesting one to look at the distribution of goblet cells within the small intestine. They increase in number as we go from the duodenum to the jejunum to the ileum. Of course, as the contents of the small intestine migrate down, more and more material gets reabsorbed, and we get whatever left over that eventually becomes feces. So it needs a little bit more help getting through the gastrointestinal tract to reach a peak within the large intestine. Anyway, I hope you enjoyed that, and I'll see you for the next one.